morning. Now we will start discussion on a new module, energy production from organic waste through anaerobic digestion. In the previous modules we have discussed on different thermal routes for the conversion of waste to energy, and in this module we will start discussion on the biological route for the conversion of waste to energy. Among different biological routes for the waste to energy conversion, anaerobic digestion is one important route, and we will start with this first. So, in anaerobic digestion, organic compounds are digested in absence of oxygen in the presence of microorganisms and produces different products, out of which the biogas is the most important product. So, depending upon the nature of the organic compounds, the complexity of the process will increase and the flow sheets will also be changed to some extent, and in this process number of steps are involved and number of microorganisms are responsible for those different types of reactions. Different pathways are applicable to get the output of this process, and there are many factors which influence the performance of this anaerobic digestion process. Type of reactors also influences it. So, all those things we will cover here in this module, and we will discuss on the anaerobic digestion of biogas production, mechanism of anaerobic digestion, microorganisms for anaerobic digestion, then pathways for anaerobic digestion, then pretreatment of lignocellulosic biomass and wastes. So, lignocellulosic biomass and waste are a special type of wastes which is getting more interest nowadays, but having some difficulty for processing through these anaerobic digestions. So, pretreatment is required and we will discuss on these things. And next, the flow sheet of anaerobic digestion of wastes, type of anaerobic digestion process, kinetics of methane formation, then anaerobic digester and their types, operation of anaerobic digester, two stage digester and processes, and then impurities in biogas and their impacts, and finally we will also discuss on sludge volume in a digester. Now, let us see the first the anaerobic digestions and biogas production. So, what is anaerobic digestion? It is a series of biological processes in which microorganisms break down biodegradable material in the absence of oxygen. So, in anaerobic conditions microorganisms will react on organic substrate and the organic substrate will be converted to biogas. This is the ultimate process, but this is not so simple and not a single step process. It is a multi steps and a complex process. It involves number of microorganisms, number of mechanisms or pathways, etc. Now, we will see what are the feedstocks that can be used in anaerobic digestion. So, anaerobic digestion can use energy crops, it can use animal waste, it can use industrial wastes, it can use MSW, municipal solid waste, it can use restaurant food industry wastes, it can use sludge from waste treatment plant. So, these are the possible important feedstocks, and what we can get from this? We can get biogas, we can get hydrogen, we can get biofertilizer, we can get compost, and we can get reusable water. So, out of these feedstocks, the animal waste, particularly cow dung to biogas, that is gobar gas, this is a very known technology, but use of energy crops, industrial soils and other these things through this route is not very well developed, particularly the energy crops and MSW, some part is having lignocellulosic biomass. So, this or waste, so lignocellulosic wastes and lignocellulosic biomass have good potentials and in terms of availability good potential, and if we can improve this anaerobic digestion, so those can also be processed through this route. Now, if we think about the primary products, the biogas, basically it contains methane. So, this is will be having some heating value, and as on today in basically the gobar gas, this is used for domestic application, the domestic cooking. but not in very big scale plant we are having, but small scale plant we are having. So, apart from this we can get hydrogen through this route also, but this technology, this is not very matured one, and people are working how to get more hydrogen through these different routes of microbiology. And then biofertilizer. So, when the anaerobic digestion reaction is going on, number of reactions are taking place and some products are formed which are nitrogenous in nature and which are having good fertilizer values, and the residual part after biogas production, that can be used for composting. So, compost can be formed, and in this process if we compare the thermochemical or thermal route with this biological route, the basic difference is that in this route we use huge amount of, very large amount of water. So, solid content in this anaerobic digestion unit for a typically for a conventional anaerobic digester, it is around 8%. So, it is around 8% only. So, remaining 92% is liquid, but if we use thermal plant as a thermal route, then the moisture content should be very less. So, most of the say around 80% should be say solid content. So, in this case we get large amount of slurry. So, that slurry contains significant amount of water, that water has to be removed or has to be reused, otherwise this process will not be acceptable on environmental point of view. That is why people are trying to improve this process in terms of low requirement of water. So, 8% slurry which is conventionally used here for anaerobic digestion, in wet anaerobic digestion, that is going to be upgraded to dry anaerobic digestions using up to 20% of slurry, 20% of solid in the slurry. So, these are the feedstocks and these are the primary products which we can get through anaerobic digestion route. Now, we will see what is the biogas. So, biogas is produced through the anaerobic digestion from the organic materials, and this biogas contains significant amount of the methane, that is 50 to 75% methane, and rest is carbon dioxide 25 to 50%. Apart from these two major components, it may also contain nitrogen 0 to 10%, it may also contain hydrogen 0 to 1%, and it may also contain H2S hydrogen sulfide, that is 0 to 3%, and in some cases oxygen 0 to 1%. So, these are the typical composition of the biogas, and here we see H2S has to be separated. So, these separations we have discussed, nitrogen separation we have discussed in the earlier modules. So, this is the biogas. So, as methane is higher, it can be used as a source of energy. Now, we will see different types of feedstocks or wastes which are available with us, and what are the potential of these waste in terms of solid content, in terms of biogas potential, in terms of methane content in the biogas. So, this table shows us that with the variations of these feedstocks, these values also vary significantly. So, in this 12.5% solid to 86.5% solid we are getting in this case dry matter, but in spite of having this very high dry matter, in case of wheat straw it is giving 367 liter per kg substrate potential of biogas production, whereas you see grass it is of only 16, but it is giving us 557 liter per kg of substrate that production. So, this indicates that although waste dry matter content is required, but it is dependent on the type of matters which is present in it, compounds present in the dry matter. So, if it is a lignocellulosic biomass, so lignocellulosic biomass is having good amount of lignin. So, if say 20 to 30% or 25% of lignin is present, so that is not useful, and it also requires some pretreatment due to the presence of cellulose, hemicellulose. So, that process becomes slower. So, and also that is why maybe the reason that it is giving the lower biogas potential than this. We will see some other examples here, that is beet leaves. poultry wastes, cattle manure, horse dung, sheep dung and pig manure. So, these are different wastes which are having different dry matter content and also different biogas potential and also the different methane content in the biogas. So, if we see the cattle manure, this is one typical applicable for gobar gas plant. So, here cattle manure cow dung around 14% solid, that is dry matter is present in it. It gives 260 to 280 liter per kg substrate and 50 to 60% of methane. In the first slide we have given that the biogas contains around 50 to 75% of methane, but here we are getting say 50 to 60% and somewhere even more than 75% like say 84 like this, somewhere 84 like this. So, these are not used in commercial scale, but efforts are going on to develop some technology for the applications of these feedstocks in commercial scale also, just the improvement of this anaerobic digestion process. Now, we will see the different steps which are responsible for the whole process of anaerobic digestion. So, if we think about the organic compounds present in it, those are complex in nature. If we exclude the lignocellulosic biomass, then other type of organics basically those are either carbohydrates, fats or protein types. So, carbohydrates, fats or proteins, those are complex molecules. So, those molecules need to be broken down to their monomers or the smaller molecules, and that is done by the hydrolysis. So, hydrolysis is the first step for the anaerobic digestion, through which those complex organic compounds are converted to simple organic compounds like sugar, amino acids, peptides, etc. So, then simple organic compounds in next step through acidogenesis is converted to short chain fatty acids. So, this acidogenesis step also, the short chain fatty acids also include some acetic acid, and it is a fermentation process. The whole process is basically based on fermentation process. So, carbon dioxide is also produced. So, carbon dioxide, hydrogen, acetic acid and short chain fatty acids apart from acetic acid are produced through this acidogenesis step. So, after acidogenesis step, the short chain fatty acids can be converted to acetic acid through acetogenesis or can be converted to H2 and CO2 through hydrogen producing acetogenesis bacteria. So, these short chain fatty acids can be converted to acetic acid acetogenesis or can be H2 and CO through hydrogen producing acetogenetic bacteria. So, this is both these are under acetogenesis step, and this H2 and CO2 further react and forms acetic acid. So, this is another type of microorganism responsible for these reactions. Hydrogen and CO2 will react and give us acetic acid or acetate. Then once the acetic acid and acetate is formed, then the next step starts to work, that is methanogenesis. So, in this methanogenesis acetic acid or acetate is converted to methane, or these some microbes are also available which directly converts hydrogen and CO2 to methane. So, these are the different types of reactions which takes place during the total anaerobic digestion process. This is for organic compounds, not lignocellulosic biomass, or if lignocellulosic biomass and lignocellulosic wastes are there. So, the process is more complex and we need some pre-treatment step before this. So, carbohydrates, fats and proteins, these materials can be produced from basically the cellulose, hemicellulose etc. can be produced from this pre-treatment process. So, this is additional step required for handling lignocellulosic biomass through this anaerobic digestion route. Now, we will see what are the contribution of different processes or different steps, that is typically how much contributions of different steps are there on the overall AD process or anaerobic digestion process. So, here we are considering organic compounds. So, organic compounds are present, not lignocellulosic biomass. So, organic compound is present here. So, that will be converted to hydrogen acetic acid and short chain fatty acid through acidogenesis and fermentative route. So, 76% of the organic compounds is converted to short chain fatty acids, 4% is to directly to hydrogen and 20% is to acetic acid. So, this is the fermentative route. So, these are the contributions 76%, 4% and 20%. So, if we have total 100% here, so 76% we are getting short chain fatty acids. Out of this 76% of short chain fatty acids, 24% short chain fatty acid is converted to hydrogen and 52% short chain fatty acid is converted to acetic acid, and there is some interchange, the hydrogen plus CO2 react to form the acetic acid, and then this acetic acid gives methane. Around 75% of methane produced is through the acetic acid and around 28% is produced from the hydrogen. So, this is 28 or round figure 30 and this is say 70. So, 70 and 30, this is the ratio of the methane produced through these two routes, acetic acid to methane and hydrogen to methane. So, hydrolysis and acidogenesis, this is the step. Next is acetogenesis and dehydration, this step is, this is the hydrolysis and acidogenesis and this is the methanogenesis step. So, these are the steps and these are the relative contribution on the overall process. So, this is the typical values, it may differ from case to case. Now, elementary description of anaerobic digestion. So, we have discussed in the previous modules also that to understand a process and to predict the products distribution, we need to express the mechanism the phenomena in terms of some equations. So, that is stoichiometric equation is required, but in this case anaerobic digestion the substrates are different, may be different and microorganisms may vary, reactors may different. So, it is very complex systems and this complex systems it is very difficult to exactly express the stoichiometric equation. So, people tried to express it on the basis of experimental data and some empirical correlations was developed and proposed in 1991 and that has been provided here, that is the biogas production, that is CH4 and CO2 production takes place from organic substrate, say organic substrate having a CHNS analysis or elemental analysis. So, CnHaObNc. So, this is the molecular presentation of the formula of the organic waste or the organic material which you are using. So, that will be available in water. So, that water will be say in higher content, say this concentration is only say 8% or say 10% and this is say around 92 or 90%. So, this percent H2 and this is our organic materials. So, people tried and shown that this relationship CnHaObNc plus 2n plus C minus B minus 9Hd by 20 minus Ed by 4 to H2O in presence of microorganisms, here microorganisms will gives up D by 8 CH4 plus n minus CHD by 5 minus D by 8 CO2 plus Hd by 20 C5H7O2n. So, this is nothing but the empirical formula of bacterial dry mass. So, bacterial dry mass will be produced in the media in the system plus C minus Hd by 20 NH4 plus. So, NH4 will also be available in the slurry plus C minus Hd by 20 into HCO3 minus. So, this is the formula, empirical formula proposed by these researchers. Now, it is interesting to see here that the whole equation includes N, C, B, Hd in the coefficients of this. So, what is that N, C, B are here we are getting from CHNS analysis N, A, B, C we can get, but two important thing, that is S and E. S and E has also been incorporated in the expression. So, what is that S? That is the fraction of waste converted into cells. The waste or organic material which is used as a feedstock, part of it is converted to bacterial biomass and part of it is converted to energy. So, that S is the fraction of waste converted to cells and E is the fraction of waste converted to methane for energy. So, if we take S plus E, that will be 1. So, S plus E is equal to 1. So, this is the empirical formula and here we are getting one C5H7O2N, that is the empirical formula of the bacterial dry mass, and CnHaOb and Nc is the empirical formula of the waste being digested. So, this is the empirical relationship or the stoichiometric expressions people have proposed for the anaerobic digestion. Now, we will see some factors which influence the anaerobic digestion process. Some important parameters like say solid content in feed wastes, that we have just discussed, that typically 8% is maintained in conventional anaerobic digestion methods, but efforts are on to increase this to around 20% for dry anaerobic digestion. C by N ratio, carbon and nitrogen ratio is also important, because nitrogen is required for the growth of the microorganism. So, CN ratio also influence the rate of reactions and the gas productions. Temperature is important because the microbes are sensitive to temperature, different types of microbes are able to grow in different temperature range. So, different types of microbes will be used, different temperature range will be used, we will discuss in later on. And pH of the solution, that is also important because that will influence the growth of the microorganisms. Microorganisms cannot sustain all pH. So, pH has to be selected on the basis of the suitability of the microorganisms. And retention time, because this is the biological methods. So, microorganisms degrade or reacts on the substrate. So, this is a very slow process. So, retention time is very high in this system, and retention time play significant role. If we cannot choose a retention time in a proper way, so we may not get the efficient use of the system. And type of digesters, there may be some other types, maybe 1 step, 2 step, okay, we will discuss later on. So, types of digesters also influence the performance of the process, and microbes types, what type of microbes we are using. Just we have seen different types of steps are there. So, different microbes are responsible if we do not use one particular type of microbes. So, obviously that phase will be stopped or will be less. So, our overall efficiency of the process will be affected. Now, we will see the groups of biogas microbes. So, what are the groups? Our objective to get methane. So, one will be methanogenic or methane forming microorganisms, and another is that is non-methane forming microorganisms. So, the initial steps are not methane forming. So, the initial steps will be responsible for the non-methane forming microorganisms, and the different steps are hydrolyzing and fermenting bacteria and then acetogenic bacteria. So, hydrolyzing and fermenting bacteria and acetogenic bacteria are under non-methane forming, and these bacteria hydrolyzing and fermenting obviously acetogenic and hydrolytic. So, hydrolytic, these microorganisms will help to hydrolysis, and acetogenic will help to convert the sugars to acids, and then acetogenic bacteria will be long chain, small chain acids will be converted to our acetic acid and acetates and hydrogen. So, these are two hydrogen producing acetogens, means acetic acid plus hydrogen will be formed, and in other case hydrogen plus CO2 will be converted to acetic acid, that is homoacetogens. And methane forming, that is acetoclastic acetate degrading bacteria. Acetoclastic means acetate will be degraded to form CH4, and another is hydrogenotropic, hydrogen utilizing bacteria, hydrogenotropic. So, hydrogen plus CO2 will be reacting and give us CH4. So, these are the different types of microbes which are used in case of AD, anaerobic digestion process. So, what the microbes work, what type of work they do on this. So, first step, that fermentative bacteria, those work on cellulose to decompose bacteria, there are cellulose decomposing bacteria, that may be protein decomposing bacteria, may be fat decomposing bacteria, and they decompose the cellulose, forms monomers, oligomers and saccharides. The protein decomposing bacteria forms amino acids, and fat decomposing bacteria fatty acids and alcohol, and all those compounds in the later stage is converted to short chain organic acids, aldehydes, hydrogen and CO2. So, this is fermentative including acidogenic bacteria. So, these are the activities of this bacteria, role of this bacteria, and the second step, that is acetogenic bacteria. The first step, the acids which is formed, that is converted to acetic acid and H2CO2, and the third is methane, that comes methane CH4 and CO2. So, now we will see what are the fermenting and hydrolyzing bacteria. So, as you have discussed that this hydrolyzing bacteria at first reacts on the complex organic compounds, and different organic compounds are there. So, those complex compounds will be converted to relatively smaller molecular weight compounds, and oligosaccharides, the polysaccharides to oligosaccharides, and monosaccharides, and proteins to peptides and amino acids, triglyceride to fatty acids and glycerol, and nucleic acid to heterocyclic nitrogen compounds, ribose and inorganic phosphate. So, these are the conversion, the first step, and we will see the relative anaerobes of genera like Streptococcus and Enterobacterium, that is Bacillus, Cellulomonas and Mycobacterium are some important hydrolytic bacteria. So, these are some important hydrolytic bacteria which can hydrolyze the organic complex organic compounds. Now, we can use the enzymes extracted from these microorganisms in place of the whole microorganism cells. So, those enzymes are cellulases, hemicellulases, amylases, lipases, proteases, and from these bacteria we can get, and they depolymerize the carbohydrates, lipids and proteins to soluble molecule. And these are the different complex compounds, and these are the converted forms through the acetogenic bacteria: carbohydrates to sugars and alcohols, cellulose to glucose and cellulose. It may be say hemicellulose. So, it is hemicellulose to say glucose, xylose, and arabinose. So, those will be the product if feedstocks is hemicellulose. Now, proteins, amino acids, peptides to fatty acids, and lignin. Lignin is not degraded by the microorganisms, and hydrolyzing after then we are coming to your acetogenic bacteria. So, some examples of this acetogenic bacteria are like this: the bacteroids, Bifidobacterium, Pretreatment, Eubacterium and Lactobacillus. Those microorganisms act as acetogenic bacteria. Apart from this, some bacteria like coliform bacteria like Escherichia coli, Salmonella and Shigella. So, these microorganisms also react as acetogenic bacteria, but these bacteria, these microorganisms can produce formic acid, and you see the sugar can be converted to fatty acids, alcohols to fatty acids, amino acids to fatty acids, glycerol to acetate and CO2. So, these are the converted product by these microorganisms. Now, we will see acetogenic bacteria. Acetogenic bacteria we have seen two types of, that is your hydrogen producing acetogens and another were homoacetogens. H2 plus CO2 is converted to CH3COH by these microorganisms. Clostridia and Acetivibrio are some examples of this acetogenic bacteria, and propionic acids, propanols, butyric acids are converted to acetic acid by these microorganisms. But in this case hydrogen is formed, and in the second case this hydrogen and CO2 reacts to give us CH3COH. Now, we are coming to methanogenic bacteria. So, methanogenic bacteria we have seen that acetic acid to methane and CO2 plus H2 to methane. So, this is called acetoclastic methanogens, and another is hydrogenotrophic methanogens. So, these are the reactions here. So, this is a 70% and this is a 30%, but these microorganisms, these are highly sensitive and having slowest growth rate, and these microorganisms are less sensitive and having higher growth rate. So, apart from this, the methane can also be formed from formic acid and methanol, they can give the CH4 also. So, some microbes will be responsible for this. On the basis of this, the methanogenic bacteria are classified into three categories. So, depending on their 16S ribosomal RNA or rRNA and their substrates used and growth and methanogenesis. So, they are grouped, just we have discussed the 3 groups 1 to 3. So, these 3 types of microorganisms are available. The first groups, so methanobacterium and methanobrevivector, and the second is methanococcus, and the third group generally includes methanomicrobium and methanogenium and methanospirillum and methanosarcina. So, these are the different types of microorganisms which are responsible for the methanogenic phase, and this is the pathway through which the acids are formed. So, EMP pathway, that is embedded Marhof and Parnas pathway, this pathway explains the production of intermediate product pyruvate from the sugar. Then pyruvate is further converted to different acids, and depending upon the partial pressure of hydrogen in the systems, acetate and other higher acids are formed. So, if the hydrogen pressure is lower, acetate is favored. If higher hydrogen is there, so partial pressure of the hydrogen is higher, then propionate, butyrate, ethanol and lactate are formed. So, and we have already discussed that there is also a special mode of cleavage of intermediate pyruvic acid to formic acid in presence of E. coli bacteria or enteric bacteria, and this is the pretreatment for lignocellulosic biomass and waste. As we have discussed that non-lignocellulosic biomass and waste can be converted through the routes which we have discussed just now, but if we have lignocellulosic biomass, we need one pretreatment step. We have to convert cellulose and hemicellulose, we have to remove the lignin part from it, and cellulose and hemicellulose has to be converted into monosugar. So, that for that purpose physical pretreatment, chemical pretreatment or chemico-physical pretreatment and biological pretreatment methods have been applied, and these physical pretreatment methods include cell milling, some irradiation and some other methods, that is hydrothermal and pyrolysis method. So, these are the 3 major important methods which have been used under pretreatment method under physical pretreatments, and these are the possible changes in the biomass. This decrease in cellulose, decrease in degrees of polymerization, increase in accessible sugar, and these are the notable remarks. Most of the methods are highly energy demanding and most of them cannot remove lignin. So, this is not suitable one for the lignocellulosic biomass, but this is more suitable one chemical, it can remove the lignins, and this can be done by alkali treatment, by acid treatment, by gas treatment or reaction with some oxidizing agent, and it decreases the cellulose crystallinity, decreases degree of polymerizations, and these methods are among the most effective and include the most promising process for industrial applications for the pretreatment of the lignocellulosic biomass. Another is biological pretreatment methods, there are some fungi and actinomycetes have been used, but these methods is very slow method and delignification can take place, but due to the slow rate of reactions, these are not getting that much of interest. So, up to this in this module, and we will discuss the rest part in the next module. Thank you very much.